Tantor Audio, a division of Recorded Books, presents The Family Tree Guide to DNA Testing and Genetic Genealogy, 2nd edition, by Blaine T. Bettinger, narrated by Mike Chamberlain. Publisher's Note: This audiobook includes bonus material. Please visit Tantor.com and type The Family Tree Guide to DNA Testing and Genetic Genealogy into the search box. Follow the link to the book's page. Then, under the Supplemental Contents section, please enter the access code RELATIVE and then click the Unlock button to access the bonus material. The code should be entered in all lowercase letters. All images, tables, examples, glossary, and appendices referenced in this audiobook can be found in the bonus material. Introduction. Genealogical records are not perfect. Our ancestors had poor memories, just like we do. They bent the truth to make themselves younger or seem more favorable, just like we do. And they made up stories just like we do. In addition, genealogical records can be altered, improperly transcribed or recorded, or completely lost, even in the years immediately after their creation. As a result, genealogists work with a trail of imperfect and inconsistent breadcrumbs, and we use these traces to recreate the lives of our ancestors. Sometimes we do a good job, sometimes we do a poor job, and sometimes we may not know the difference. Trapped within your DNA, however, are the stories of your ancestors. Although this information was inaccessible to previous generations of genealogists, modern genetic testing has allowed us to extract those stories and begin to add them to the rich genealogical tapestry so many of us spend our lives creating. Although DNA is a mostly unchangeable record of those ancestors who provided bits and pieces of their DNA to the current generation, we are currently limited in our ability to properly interpret the whole record. Indeed, interpreting DNA test results can introduce errors and inconsistencies. As a result, current genetic genealogy testing is not yet a perfect genealogical record either. Only when DNA and traditional genealogical records are combined do we begin to fully extract the full value of genetic testing. My Genetic Genealogy Journey. I was introduced to genealogy in the 7th grade. My English teacher assigned a short homework assignment, a 4 or 5 generation family tree that we were to fill out as far back as we could by asking family members for information. My parents recommended that I call my grandmother, as she was one of the oldest members of that generation. During the call, my grandmother recited a litany of names and places completely from memory. I frantically filled out the paternal side of the family tree, then added sheets of paper with even more ancestors. With that one phone call, I was hooked. I've spent the ensuing 25 years trying to verify those names, learn about those ancestors' lives, and fill in the blanks that my grandmother couldn't provide. I took my first DNA test in 2003. I was a biochemistry graduate student in Syracuse, New York, and a DNA test was the perfect marriage of the two great loves of my life, genealogy and science. Unlike most other people at that time, who started their DNA journey with a Y-chromosomal, Y-DNA, or mitochondrial DNA, mtDNA test, my first test was an autosomal DNA, atDNA test. It only looked at a little over 100 markers, compared to the hundreds of thousands of markers used today, but I was once again hooked. That autosomal test was just the first in a very long line of DNA testing, including a full sequence of my entire genome by the Personal Genome Project 10 years later. Along the way, I've learned a great deal about my genealogical heritage as a result of DNA testing. I know that my mitochondrial DNA has indigenous, meaning that my mother's 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 mother was indigenous at some point. I know that I carry African and indigenous segments of DNA from Central American ancestors. I know that I have pieces of DNA passed down to me from my French-Canadian and Irish ancestors. And I am beginning to identify the parents of my adopted great-grandmother, all due to DNA testing. I've used modern genetic testing to extract stories that I was carrying with me all along, stories that my ancestors, including the grandmother who gave me the gift of genealogy, unknowingly passed down to me. Across the country and across the world, every vial of spit and every cheek swab thrown in the mail with anticipation is overflowing with long-forgotten stories waiting to be unlocked from within a series of A's, T's, C's, and G's. How to use this book. This book is intended to be a resource for genealogists of all experience levels, from beginner to expert and everyone in between. If you've never taken a DNA test, then you can use this book as a primer to understand what DNA testing is, whether DNA testing is right for you, and how you can use the results of DNA testing to examine your ancestry. My advice is to listen to the book from start to finish as it is presented, since it is written with more basic information at the beginning and more advanced information toward the end. Throughout the book, you'll note special terms indicated in red. These are important vocabulary words you'll encounter in your genetic research, and I've compiled them into a glossary toward the back of the book for your reference. If you've already taken a DNA test, you can use this book as a reference while you review your test results. After making a quick stop at chapters 1, Basics, 2, Common Misconceptions, and 3, Ethics, turn to the chapter relevant to your type of DNA testing. AT-DNA, chapters 4, 8, and 9, Y-DNA, chapter 5, MT-DNA, chapter 6, and X-DNA, chapter 7. Then, listen to the remaining chapters to ensure you've filled in all the blanks and have a complete understanding of the different aspects of genetic genealogy testing. A genealogist's education is never complete. It is important that genealogists stay on top of the latest developments in DNA testing and analysis. Accordingly, if you feel you've mastered most of the topics presented in this book, turn to the More Resources section in Appendix C for links to some of the best blogs, forums, and mailing lists available. These links will allow you to discover and explore all the latest developments and advances in genetic genealogy. What's new in this second edition? The first version of this book was published in the fall of 2016. Autosomal DNA testing was exploding in popularity in the United States and several other countries, and there was a need for education as millions of people received their test results. The Family Tree Guide to DNA Testing and Genetic Genealogy met that need and quickly became the fastest-selling book in the field. However, as with most areas of modern genealogy, the field of genetic genealogy is changing quickly. In the few years since the first version of the Guide to DNA Testing was written and published, new tests, testing companies, methodologies, and third-party tools have all been introduced. DNA test takers have more choices and opportunities than ever before. But with more selection comes a greater need for education. This second edition of the Family Tree Guide to DNA Testing and Genetic Genealogy will help you navigate these latest developments and will help you uncover the history hidden in your DNA. New and updated sections of this book cover new tests and tools such as MyHeritage DNA, myheritage.com/DNA, Living DNA, livingdna.com, and DNA Painter, dnapainter.com, as well as the big Y700 test from Family Tree DNA, familytreedna.com. This book also covers DNA features and topics that have come to age since the first edition's publication in 2016, including shared matching, ethical questions about the use of DNA information by the government or other third parties, xDNA advancements, and much, much more. I wish you the best of luck as you embark on your own DNA journey. Blaine T. Bettinger, October 2018. Part 1. Getting Started. 1. Genetic Genealogy Basics. Genealogists are family historians, documenting known information about a family and using historical records to recreate and recover information that has been lost due to time and distance. As a result, personal possessions like family Bibles, wartime letters, and dusty daguerreotypes are among the most treasured items a genealogist can receive from an ancestor or relative. These possessions are often unique and reveal information that may otherwise be lost. Passing down these treasured family records and keepsakes is an important tradition that preserves memories for future generations. However, prior generations have been passing down more than memories and keepsakes to their descendants. At every generation, our ancestors passed down indelible records through their DNA, pieces of themselves that they received from their own ancestors. That inheritance is the reason you have your great-aunt Millie's curly hair, your grandfather's heavy eyebrows, or your great-grandmother's deep blue eyes. You are the keeper of your ancestors' DNA, and with a new tool called genetic genealogy, you can unlock that DNA and reveal the secrets it has safeguarded for generations. Indeed, even adoptees who have no knowledge of their biological ancestors can use this tool to find genetic relatives and learn about their biological heritage. In this chapter, we'll outline the history and basics of genetic genealogy to prep you for the rest of the book. As you listen, you'll learn more about the different types of genetic genealogy testing and how you can use your results to examine your heritage, answer genealogical questions, and solve family mysteries. You'll learn about which tests you should take and what limitations you should keep in mind when reviewing your test results. You'll also discover, among many other things, some of the third-party tools you can use to wring every bit of useful information from your DNA test. The history of genetic genealogy. Before genealogists used it, genetic genealogy was utilized by scientists and historians to identify genealogical connections between high-profile historical figures. In 1994, for example, mitochondrial DNA, mtDNA testing, one of the first tests to become available, was used to identify skeletons found in 1991 in a shallow grave in Yekaterinburg, Russia, as those of the Romanov family who were killed in 1918. Using a low-resolution test, scientists discovered that mtDNA extracted from several of the skeletons, including those hypothesized to be the Tsarina Alexandra, a maternal granddaughter of Queen Victoria in Image 1A, and several of the Tsarina's children, matched mtDNA obtained from Prince Philip, Duke of Edinburgh, who was a great-great-grandchild of Queen Victoria. Since that early testing, both mtDNA testing and autosomal DNA, atDNA testing, another popular type of testing, have identified the remains of Tsar Nicholas II and his entire family, including the Tsarina and all 5 of their children. In a similar way, Y-chromosomal, Y-DNA testing was used in 1998 to show a genetic match between a male relative of President Thomas Jefferson and a descendant of Eston Hemmings, the youngest son of Thomas Jefferson's slave Sally Hemings. The descendants of Eston Hemmings had a strong oral tradition that Eston's father was indeed Thomas Jefferson, and Eston was said to bear a strong resemblance to Jefferson. Many historians, however, believe that Eston's father was one of the sons of Jefferson's sister, which might explain the resemblance. Since the former president had no surviving legitimate sons to pass down Y-DNA to, researchers obtained a Y-DNA sample from 5 male-line descendants of Jefferson's paternal uncle, Field Jefferson. Those samples were compared to Y-DNA samples obtained from a living descendant of Eston Hemmings, and the two were a genetic match. Today, most historians accept that Jefferson fathered several children with Sally Hemings, including Eston. Recognizing the power of DNA to examine genealogical relationships, genealogists began to investigate ways to use the tool. A few years after historians successfully used DNA testing to reveal Jefferson's descendants, a group of scientists, including a man named Brian Sykes, conducted a study examining the Y-DNA of 48 males in the United Kingdom with the last name Sykes. The low-resolution Y-DNA test determined that almost half of the males were related through their paternal or surname line, suggesting a single surname founder for these males. The scientists noted that Y-DNA studies, such as the one they had conducted, could have numerous applications in forensics and genealogy. Eventually, the practical implications of DNA testing for genealogists became clear. In early 2000, 2 companies began offering DNA testing to genealogists. Family Tree DNA, FamilyTreeDNA.com, based in Houston, Texas, and led by Bennett Greenspan, Max Blankfeld, and Jim Warren, and Oxford Ancestors, OxfordAncestors.com, based in Oxfordshire, England, and created by Brian Sykes of the Sykes Surname Study. Both companies launched by offering Y-DNA and mtDNA testing to genealogists, the first such commercial products. Over the next few years, genetic genealogy testing expanded widely, led by large projects that combined Y-DNA testing and surnames, similar to the Sykes study's methodology. In the fall of 2007, genetic genealogy testing company 23andMe, 23andMe.com, began offering the first commercial AT-DNA test. Ancestry DNA, Ancestry.com/DNA, launched its own AT-DNA test 5 years later in 2012, and MyHeritage DNA, MyHeritage.com/DNA, and LivingDNA, LivingDNA.com, both launched AT-DNA tests in 2016. Today, these companies offer genetic testing to genealogists of all experience levels and are the leading genetic genealogy companies. We'll learn about these and other testing companies in a later chapter. Charles Darwin and genetic genealogy. In the mid-1800s, Charles Darwin first proposed the groundbreaking theories of evolution and natural selection, upon which much of modern genetics is based. Nearly 200 years later, Darwin's own genetic roots were examined with a simple DNA test. In early 2010, National Geographic's Genographic Project tested the Y-DNA of Darwin's great-great-grandson, Chris Darwin of Australia. The test revealed that Chris, and thus most likely Charles, belonged to the R1b haplogroup, the most common haplogroup in males of European descent. We'll discuss haplogroups in more detail later. Genetic genealogy today. Genetic genealogy is an essential tool for genealogists. It is an important piece of evidence similar to a census record, will, or land record, and it might be the last piece of information available in locations where records have been lost or destroyed. Although DNA testing cannot answer or even shed light on every question, savvy genealogists should at least consider it as part of every genealogical research project. In the summer of 2015, 23andMe and Ancestry DNA each announced they had tested their 1 millionth customer, and their customer base is growing, with thousands of new tests being sold each month. Although the Family Tree DNA database has traditionally been smaller than 23andMe's and Ancestry DNA's databases, it is undeniably large and continues to grow rapidly. Additionally, with MyHeritage and LivingDNA also offering testing, the number of people that have taken a genealogy DNA test has easily exceeded 20 million at the time of publication and continues to grow by leaps and bounds. As the database's sizes grow, so does the power of genetic genealogy. New connections, tools, and discoveries will be made possible as more and more people take DNA tests. A little genetics. What is DNA? You don't need to have an advanced degree in molecular biology or genetics to understand genetic genealogy. You don't even need to remember anything from that biology course you took in the 10th grade. This brief introduction and some details provided in each individual chapter will be more than enough to help you understand how to use genetic genealogy testing for your research project. The cell, the basic unit of life, uses genetic material called DNA to control the vast majority of its functions, beginning with the division of its parent cells and ending with its ultimate death. DNA, short for deoxyribonucleic acid, is a component of the cell that carries instructions for the development and operation of all living things. A small percentage of the DNA comprises genes, short segments of DNA that are used as the blueprints to create a protein or an RNA ribonucleic acid molecule. Scientists also continue to find secondary functions for the non-coding regions of DNA, which don't specifically create proteins or RNA. A molecule of DNA is composed of a string of millions of smaller units called nucleotides. Together, two intertwined DNA molecules interact to form a single double helix structure called a chromosome in the nucleus or control center of the cell. A normal human cell has 92 long molecules of DNA that pair up to form 46 double-stranded chromosomes. Each of these, in turn, forms a chromosome pair with another similar but not identical chromosome to create 23 different chromosome pairs. Confused? There's a table in the bonus PDF that breaks down the different levels of organization of DNA. In addition to the DNA in the nucleus, hundreds or thousands of copies of a very small circular strand of DNA are found in the many mitochondria outside the nucleus. Mitochondria are tiny powerhouses of the cell responsible for, among other things, creating the energy our cells need to function. Image 1b, a karyogram, is a photograph of a human's karyotype, which is all of the chromosomes of the human cell arranged in pairs in a numbered sequence from longest to shortest. To make a karyogram, researchers stain chromosomes with a special chemical, then take a photograph of the stained chromosomes. The chromosomes are then digitally rearranged into pairs and organized into a specific numbered sequence. This karyogram also includes a ring of mtDNA for reference. In this book, we will examine the 4 types of DNA used for genetic genealogy: atDNA, yDNA, mtDNA, and xDNA. Autosomal DNA, atDNA, is composed of pairs of chromosomes found in the nucleus of the cell. Humans have 23 pairs of chromosomes, 46 total, of which 22 are autosomal DNA, or autosomes, and 1 is yDNA and or xDNA, sex chromosomes. See the respective sections. 1 copy of each chromosome is inherited from the mother and 1 copy from the father. An atDNA test reveals information about both paternal and maternal lines, and most commercial tests examine this kind of DNA. We'll discuss this test more in Chapter 4. Y-chromosomal DNA, yDNA, focuses on the Y chromosome, one of the 2 sex chromosomes that determine gender, the other being the X chromosome. Only men have a Y chromosome, and a yDNA test reveals information about the male test taker's Y chromosome, which is exclusively passed from fathers to sons. We'll go into more detail about the yDNA test in Chapter 5. Mitochondrial DNA, mtDNA, is a small, circular piece of DNA found in the cell's energy factory, the mitochondria. This is the only DNA not found in the cell's nucleus. mtDNA is passed exclusively from mother to child, and an mtDNA test reveals information about the test taker's direct maternal or umbilical line. Chapter 6 focuses on mtDNA testing. X-chromosomal DNA, xDNA, focuses on the X chromosome, one of the 2 sex chromosomes that determine gender, the other being the Y chromosome. Women have 2 X chromosomes, one from their father and one from their mother. men have one X chromosome from their mother. X DNA is usually tested as part of an AT-DNA test. For men, the X DNA test, the subject of Chapter 7, reveals information about maternal lines. For women, the X DNA test reveals information about both maternal and paternal lines. 2 family trees, 1 genealogical and 1 genetic. One of the most important aspects of understanding and interpreting DNA test results is that everyone has 2 very different but overlapping family trees, one that's genealogical, reflecting familial relationships, and one that's genetic, reflecting genetic makeup and patterns of inheritance. In short, your genealogical family tree will contain everyone in your genetic family, but not vice versa. The genealogical family tree. The first, and probably best known and most studied family tree is the genealogical family tree, which contains every ancestor who had a child, who had a child, who had a child, and so on. A fully grown genealogical tree, image 1C, contains every parent, grandparent, and great-grandparent back through history. In most cases, this is the tree that genealogists spend their time researching, often using paper records such as birth and death certificates, census records, and newspapers to fill it in. Many genealogists find that the paper trail ends or becomes much more difficult to identify beyond the 1800s or 1700s, making it difficult to fill in many of the openings in the genealogical family tree. The genetic family tree. The second family tree is the genetic family tree, which contains only those ancestors who contributed to your DNA. While this overlaps with your genealogical family tree, not every person in a genealogical family tree contributes a segment of his or her DNA sequence to the test-taker's DNA sequence. A parent does not pass on all his DNA to his children, only about 50%. As a result, bits and pieces of DNA are lost in each generation. Your genetic family tree likely contains fewer ancestors than your genealogical family tree, somewhere between 5 and 9 generations back. As shown in image 1D, where green cells indicate that the ancestor provided DNA to the test-taker, and white cells indicate that the ancestor did not provide DNA to the test-taker, the genetic tree is actually just a subset of your genealogical tree. A genetic tree is guaranteed to contain both biological parents, who each contributed approximately 50% of the test-taker's entire DNA sequence. The genetic tree also likely contains each of the test-taker's 4 biological grandparents and 8 biological great-grandparents. But it is much less likely with each generation that every person in the genealogical family tree contributed a piece of their DNA to the test-taker's DNA. Since test-takers have a genetic family tree that is a subset of their genealogical family tree, a person will often share his genealogical family tree with another individual, but their genetic family trees do not overlap. This simply means they did not both inherit the same DNA from their shared ancestor. These individuals are genealogical cousins, but not genetic cousins, as genetic cousins share both a genealogical and a genetic link to one another. That is, they share one or more recent individuals in their genetic family trees, and therefore share detectable amounts of DNA from this common ancestor. Biological first cousins, for example, always share DNA, and therefore will always be both genealogical cousins and genetic cousins. Unfortunately, no one has yet been able to construct a very complete genetic family tree, due in part to the lack of extensive databases combining paper genealogy and genetics. With the recent development of tools that combine genetic testing with family trees, however, genetic genealogists are beginning to reconstruct portions of their genetic family trees. Core concepts. Genetic genealogy basics. Genetic genealogy started as a tool in history and forensic science. In 2000, DNA companies began offering genetic tests for use in genealogy. Genealogists use 4 different kinds of DNA in testing.
Sample complete. Ready to continue?